We're on slide 20. I want to move on a little further with that one.
There we are.
When we ended yesterday, we were discussing the peroxisomes and saying how much it looked like oxygen. This is a new one here that we've talked about it, but never officially like this. The mitochondria is a weird kid on the block. He is nothing like the other organelles. It's like two bags. A bag inside a bag. That's why you got a double membrane. And the inner membrane got the folds on it, and the folds are called cristae. I got a picture coming. Oh, you don't have that one?
I think I left it in my book.
Okay. And these are the ones that make energy for you. And they can make it from all kinds of organic compounds. They can even break down nucleotides and break those bonds. And when you break the bond, out comes energy. And you use that then to make ATP, and that's how you save energy. But they prefer glucose. They really like to use glucose. But if push comes to shove, they can use your own muscles. If you're somewhere you can't get food, and you start dissolving your own body. How to try to make you live longer. You break down fat, you can break down protein. But the mitochondrion has a sweet tooth for glucose. That's his favorite fuel. And the power plants, I've heard that as long as I've been around. Even when I was a student, they called them power plants way back then, back in the 70s. So that's not a new concept. I want to click it. Y'all ready? All right. Okay, I'm good. Thank you. Now, we're still discussing mitochondria now. And I'll still show you a picture. Let me look at that. I think I will. Yeah, good. Okay. The stuff inside the mitochondrion is called matrix. And the matrix contains ribosomes. And a very unique kind of DNA that's only found one place, in the mitochondria. Nowhere else in that cell would you find that. And we even call it mDNA, which means mitochondria. If you look at the DNA there, and what's in the nucleus, they're totally different. And muscle cells have a whole lot of mitochondria because they need so much energy. Do you reckon bone cells have a lot of mitochondria? No. They're just sitting around. If your job demands that you're very, very, very active, like nerve cells are and muscle cells are, you're going to have more than your fair share of mitochondria because you need the energy. Now, have I not talked to you about the theory of how mitochondria came to exist in the cell?
No.
About I thought it was a bacterium?
No.
And that would explain why the DNA is so different in the mitochondria, because if it were a bacterium, it would have been different. It's like having a black family adopt a white child. Child becomes part of the family, right? Did they ever lose those chromosomes though? No. Even though he's part of the family, fully incorporated like this is, they never lose their identity, and these don't either. And like I told you earlier, I don't understand one thing, that if I'm the cell and the mitochondria inside me are not of me, and I'm going to divide and make none of myself, who tells the mitochondria double your numbers for the new cell? I don't have a clue. I don't know how to get the words. A lot of times, you don't get words in your electrical work, or you forgot to bring something. You got to be in the loop. And I don't know how they're in the loop. I have no clue how they're in the loop. But they're in it because they double themselves too. If I need 100 to function, I make a new cell. He needs 100 too, right? Now I gotta make 200 before I actually divide, so when I do divide, my numbers are right. But I don't know how this thing knows to make a copy. These are all through, and now we don't have a scope like this. These are electron scopes, high dollar scopes. And why is this one long and that one round? Just like this. How's that shaped?
Round.
How's that shaped? That's what's happening. It's how they're laying when we take the picture. The one down at the bottom was laying like this. And the one on the top, they're laying like this. So if you ask them what's the shape of mitochondria, you can't say spherical like a tennis ball. They're elongated like this. But when you see them this way, you can't tell it because it goes back in this way, right? So anyway, but do you see all the little membranes inside? The membranes are the cristae. And all this stuff here are ribosomes. This is the matrix. That's matrix. This is the fold. And do you see right there an outer fold? You see an outer membrane right there? And the inner membrane makes these. You have one membrane wrapping around, like right here. One membrane wrapping around. Another membrane inside going back and forth. This is a crista. There's two cristae. And right there, that light color area, that's the matrix. This, if you ever take a chance to look at a bacterium, it looks like that. And that's when we began thinking that, well, the mitochondria may have been a bacteria back in the Dark Ages. That makes perfect sense. And what you see over here, anyone know what that is? It's got ribosomes stuck to it. What organelle you know of has ribosomes stuck to it?
And the plug.
The rough
ER.
ER. That's some of the rough ER in there.
So is it inside of the rough ER, or is it just right next
It's
to
just
it?
next to it. Just right next, like next to a parking lot. It's not inside. No, it's not inside. Matter of fact, those are two separate organelles, the rough ER and the mitochondria. Went the wrong way? Yeah. Centrioles. These things haven't made. They only work one time in their whole life. They are short cylinders. And if you look at one cylinder, you're going to see, let me show you what I'm talking about. All right. How many do you see? How many do you see, Emma? 3, right? Mm-hmm. All right. If I had more of these laying like this, actually I would have 9 sets of 3. And then there'd be a set here, set there, set there, set there. And they go around, they're arranged in a circle. And the picture, I'll show you the picture. See how they're arranged right there? So there's my set of 3. What is 9 times 3? How many magic markers would I need to show you that? I only got 3 right here.
A lot.
I would need how many? 27. You see 27 right there? Because there's 3 markers, which I'm showing you. I wish you had 3 more. Wish you had 3 more. And if you look carefully, this is the real picture. They're right there. I see them perfectly.
What does it do?
It's going to make that cell divide. I'm coming to there though. But I want to show you first what I mean by 3 sets 9 times. That's 3 sets 9 times. Now let me go back where we were. Okay. They lie like this. They're never parallel. They're always at right angles. They might be like crossing a T. They might be like this. They're never like a railroad track. Never. Well, there might be one somewhere. We've not found one ever in any kind of cell which are arranged side by side. They're always arranged perpendicular to each other. Your nerve cells, do they or do they not divide and make new nerve cells? Your brain cells, can you make more of them? No. You know why? They have no centrioles. You must have centrioles if you're going to reproduce yourself. If you don't have any of them, you can't. Nerve cells, neurons, they have no centrioles. The one you have right now is what you're born with. And you have when you die. So take care of it. You can't make a new one. They don't have the machinery inside. Okay? And that's why we're so worried about brain damage, because your heart can heal back and make new cells. If you have a minor heart attack, you can heal back good as new. Brain cannot heal because it makes new cells.
So when you have an accident, like a car trauma, you know, that's why when people lose their memory or something like that?
Yeah, sure is. Now, this shows you the 9 sets of 3. You do see 9, right? And 1, start at 1, 2, 3, 4, 5, 6, 7, 8, 9, back there again. And this is redrawing them. This shows you how they're arranged. Why do you see just 2 little stripes and not a circle like that? Because they're hollow. If you look back at the previous picture, if you cut it right across here, will you see these also? If I cut it right across there, my line passes through this one, passes through that one. That is deep, that is shallow. It passed through this one, there are some deeper, and there's some I removed, right? Now this one shows you all 3 of the bad boys, because I can, if I cut it this way, if I cut that thing like this, you're going to see this side and that side, right? You want to see that? You want to see that move? This way you see all of it, won't you? You're seeing there. That's how we know the shape of it. If it was solid, there'd be stuff inside of here too, right? Now there's another picture here. This is nice, buddy. Shows you the perfect shape. And there's one right angles to it, proving there's nothing in here. Because when I cut it, I can see this one, and I can see maybe those 2. But nothing in here gives you nothing in that picture. We can look at pictures and get a feel for how something's shaped. Because if it was solid inside, it'd be white all through there too, would it not? That's how we know the sucker is hollow. And the centriole, like I say, what his job is going to be is this. I have in my body right now, all my cells have 46 chromosomes. They're arranged in pairs, right? I got 23 pairs. Now, if I'm going to make a brand new cell like I am, he wants 46 too, Daddy. So before I divide, what must I do my 46? Make copies of it. I have 92 for a little while, don't I? And then as I divide down the middle, 46 stay with me, and 46 go with the other one. And when you finally get the cell divided, it has 2 brand new cells. He's got 46, he's got 46. The question is though, when a cell divides down the middle, the chromosomes better be gone. See the magician where he saws and puts spears in and the thing's sticking through it, but he's not sticking through it because she's not there really true. Well, these centrioles, They migrate both sides, they're as far apart as they can get. They send out fibers, and the fibers hook onto the chromosomes. The fiber's short, pull the chromosomes away. If you had a bowl of spaghetti, and you got one spaghetti left, and you took your fork and you pulled it, what shape would it take as you pulled it across the bowl? L or a V or what? I mean, like a V shape, right, as you pull it. Well, we now know that when you look at a cell dividing, all the chromosomes have little V shapes because they're hooked in the middle, being pulled out of the center by the centrioles. They only work one time when you got to pull the chromosomes out of the way. And they sit there the rest of the time just getting fat and happy. But when they work hard though. The chromosomes are bigger than they are. Alright? We're going to stop right there. We come in, well, not tomorrow, of course, but after we'll come in after Thanksgiving and start with slide 9.26. And I will put on Angel tonight a answer sheet. I've got to get it ready. It's not ready yet, so be watching for it to appear. Because the answers go on that sheet. It will match my key that way. I can put it back.
So you want us to print out this answer sheet?
You would have to.
Okay. The one he's going to put on Angel. Organelles.
I'll make it tonight. I'm watching football.
Okay.
Do you have a picture of the front of this